So, hello and right on time. Welcome to today's Monday seminar. We have a special guest today, a visiting researcher from University of Essex, Anna Hughes, who will introduce herself in a minute. And I hope you'll enjoy the talk. And without much further ado, Anna, take away.
Thank you very much. Can you all hear me okay? Perfect. So yes, thank you so much for all coming today. So my name's Anna and I'm a lecturer at the University of Essex in the UK, which is about an hour away from London. Nobody's ever heard of it. I'm here because of this Yeren scheme, which you might have heard of. It's a kind of mobilisation scheme amongst a range of different European universities, which both Essex and Constance are part of. So I'm basically here today and have been for a few days, just kind of getting to meet people and hear about all the cool work that's been going on in Constance and hopefully building up kind of further collaborations in the future between us and you guys. So today, I just want to give you a little bit of the flavour of some of the things I've been working on. I know a lot of you are collective behaviour specialists. I haven't done a ton on collective behaviour, but I'm hoping to explain to you how the things that I have been doing I think would be really interesting in a collective behaviour context, and these could possibly be really interesting areas for future research. So, moving on, hopefully. It's going well. There we go. So I'm a psychologist / biologist by training, and I've been involved in lots of different projects that kind of combine these areas in kind of interesting ways. And I'm interested in how humans and other animals perceive the world and make decisions about it. So I've looked at all sorts of things like why zebras might have stripes. I've looked a little bit at cuckoos and their amazing brood parasitic mimicry abilities. I've looked a bit more generally at camouflage and optimal camouflage strategies. But today I'm just going to talk about a couple of the projects that I've been working on over the last couple of years, just to give you a bit of a flavour of, sort of my interest in the psychology behind vision and how hopefully this can, inform ecological questions. So the first project I want to tell you a little bit about is the work I've done on motion dazzle. So this idea of motion dazzle, some of you might have come across. It's this hypothesis. It's a very old one. I think Abbot Thayer was the first person who proposed it in the early 1900s, that there's this idea of these high contrast stripes or zigzags or very high contrast patterns. You see them quite regularly in nature, and people have wondered why. And the classic example is the zebra, right? Why would it have these beautiful black and white stripes that don't seem to make it hard to see, and you might think would make it easier for a lion to target it? And there's been a range of different hypotheses about why this might be, but one of them is this idea that it might act to make it difficult to accurately judge the speed or direction of the zebra when it's moving. So if you're a lion trying to chase after the zebra, maybe something about the stripes is going to interfere with its motion processing system and make it difficult to catch. And interestingly, after they proposed this, the army actually got really interested in this. So there's these razzle-dazzle painted ships were used in World War I. So they went off and painted these enormous ships with these sort of lovely geometric patterns, all without really knowing whether any of it worked, to be honest, but they thought they might have a go at it. And ever since then, I think basically they decided that painting was too much effort, and so they stopped doing this. And then kind of nobody really looked at this hypothesis for a very long time. It just, kind of dropped out of, what people were looking at. So about 10 years ago now, people started to think, well, maybe we can test this hypothesis more to see whether maybe these sorts of striped targets do indeed change people's perception of the speed and direction of a moving target. So this is something that I was looking at during my PhD, and I must confess that I used humans as my subjects, so you can immediately see that this is still a long way from the natural situation, but we were working in the, the confines of this still being fairly new, and we just wanted to see, even with human participants, can you see evidence for stripes affecting judgments? So this is a really simple experiment, and basically what we got people to do was they would see a nice little stripy patch like this, and it would move along the screen in a linear trajectory, and it would disappear behind this occluder. So they could move at different trajectories. And we just asked people to make judgments of where they thought it would cross this scale bar here. So we're looking at how people's judgments of trajectory are affected by the patterning it has. And we had stripes in different orientations, so kind of vertically striped, and these 45-degree striped ones as well. And that's the main thing you need to know. I should point out that in this experiment, we did also have some of our targets also had internally moving stripes, so something a little bit like this, with the stripes kind of moving as well as the target. This is partly because in the psychology literature, we know that these sorts of targets create really strong motion and position displacement effects. So we reasoned that we should be able to see strong position displacement effects with these types of stimuli, even if not with just the static ones. What we actually find was quite interesting. So we have, these are the difference between the real and subjective crossing points. You don't need to worry too much about the numbers, but what you can see here is this is when the targets were moving from left to right across the screen. And if the stripe direction appeared to be pointing kind of upwards relative to that motion direction, people judged the crossing point as being higher than if the stripes appeared to be pointing downwards. And you can see exactly the opposite effect for when the targets were moving right to left. So we see in these sorts of kind of very controlled psychophysics experiments that there does seem to be some evidence for these stripes causing these positional kind of direction illusions. And interestingly, these are actually much stronger than the drift effect. So you can kind of see these three drift conditions just kind of form a clump. The much stronger effect is the orientation of the stripes, which we weren't really expecting. We thought that that would be a stronger effect, but it seems that just the orientation on its own was causing illusions. So this is obviously a very kind of detached, psychophysics approach to the problem. So we were trying to get a little bit closer to something that might be more like a real ecological situation. So you can run sort of little experiments where people are actually trying to catch moving targets. So, whoops, I'll just hold that, hang on. So you have, in this case, a little arena, and people are playing on, an iPad or a touch screen, and a target will appear outside from this central kind of occlusion zone in a random direction, and all people had to do in this sort of game was catch it. And we had different types of target patterning, and we did various experiments looking at this. And the basic message from all of these, and from other people who've done similar research using these kinds of experiments, basically shows that the striped ones were relatively hard to catch, but just as difficult were sort of uniformly patterned grey targets. I should say that we tend to do all these experiments in black and white, partly because colour is quite hard to get calibrated properly on these sorts of screens, and it adds an extra variable. And it's also going to be much more difficult to emulate anything like a natural situation, because the animals we're talking about do not have the same colour vision as us anyway. So we stuck to black and white for essentially simplicity reasons, because the contrast is really one of the key things that people think makes up this motion dazzle hypothesis. So, there was this kind of question of, well, they seem to be quite difficult, but not uniquely so. And this, I think, kind of one of the things we found a little bit unsatisfactory about this was that, of course, you can run many types of studies like this where you try lots of different patterns, but you're inevitably going to be quite limited in what you can test. You can maybe only test 5 or 6 pattern types, not even all different types of orientations in a single experiment. And we really wanted to test a greater variety of phenotypes. We really want to say, well, what is actually the optimal solution here? Is it stripes or is it something else? So we decided to use a genetic programming approach to try and actually efficiently sample the parameter space in order to narrow down and try and find out what sorts of patternings really are truly optimal in this scenario. So to do this, we had one of the key challenges of this sort of approach, of course, is that if you're going to get people to play a game and then it's going to, evolve some patterning over time, you need a lot of people to play your game. It is not a simple, trivial matter. You need lots of generations for this kind of evolutionary process to occur. So we got a little bit creative. And so in collaboration with a company called Foam, who kind of work on scientific gamification, we developed a little game that became a exhibition in something called the Eden Centre, which some of you might have heard of. It's a big visitor attraction in Cornwall, which is right down in the tip of the UK, where there's lots of beaches and nice things. So we basically had these sorts of screens, and all the visitors on the rainy days, which in Cornwall is a lot of days, would come and play the little game for us. So we got over 1.5 million plays on this game in the end, which did provide enough data for us to actually be able to use this kind of genetic engineering approach, a genetic algorithms approach. And it's a very similar little game to the sorts that I showed you before, but I can give you a little preview because they added some ridiculous sounds as well for us, which I think people enjoyed. So you can see it sort of moves about and people are just trying to click on it. Don't know why the sounds make it so much better, but they do. So it's very simple, and people basically did just a couple of trials. It was like 5 trials so that people didn't get bored while they were doing this for us. And essentially, without going into too many of the details, the bugs were generated by these sort of basic patterns that sort of combined together in quite complex ways in some cases to create the overall pattern at of the target. And essentially the bugs that were fittest, so were captured least quickly, survived into the next generation without any changes, and those that were least fit endured some mutations and maybe even dropped completely out of the population entirely, in order to allow the population to evolve based on this fitness metric. And so what happens? Well, this doesn't matter too much, but we also looked at whether the speed of the bugs changed things. So here we've got the different generations, so generation 0, 20 and 40. Obviously there's more generations in between, but just to give a sort of flavour. And what we found was perhaps a little bit dull, but basically everything went grey. So over time, all of our little bugs basically just got greyer and greyer and greyer. You obviously still have occasional other patterns, but they're going as much as they can to this uniform grey. So we don't seem to see any support for stripes offering this optimal protection in motion. So one thing we realised we had to do was to check that we hadn't done something wrong with our genetic algorithms, because these are quite complicated bits of code, and you always have the problem that maybe there's some kind of genetic drift, or you've accidentally set something up so that it will just all go grey inevitably, which would have been a bit of a shame once we'd got 1.5 million plays. But luckily, so we basically randomised all the data of the catch times and reran the genetic algorithms so that they had the control simulations using these randomised capture times, and we didn't see the same bias towards going to grey. So it seemed that it really was something to do with the fitness changing and preferring those grey targets. This is all a bit tiny, but I've obviously cherry-picked some examples for you there, but in the main paper, you can see all the lovely graphs of how the standard deviation of the luminance is changing over time, and the amount of striping changes over time, and things like this. So one thing we wanted to do about this was actually think, why might this be happening? Why is it that these grey stripes these grey patterns are doing well? And this is particularly interesting because if you look at the kind of modelling work in this field, you see with motion detection that basically people look at these kind of Ricard detector type models for how motion is processed in animals and in humans, actually. They think it's implemented differently, but it's the same maths. And there's this paper from a few years ago now which kind of showed that if you take a little kind of movie frame of some zebras moving and stick it through this Raikard detector, you get this lovely sort of coloured patchwork here, which is essentially all these different motion vectors. So you're getting motion signals going that way and this way and this way, and they use this to argue that these sorts of zebra stripes should give you these incredibly, strong motion signals in lots of different directions, which should be quite confusing. So from the motion modelling work, you would expect, and from the experimental work, that there should be directional signals that are hard to kind of pass for an animal or a human participant. And so we did just use an implementation of this model and just basically, we shoved each of the first generation of some of our bugs into this model and just ran a very simple trajectory on them, just something like this, and looked to see what motion signals were coming out. Nothing very complicated. So we looked at a few different measures of kind of how the motion signals might come out. So there's a couple of different things you can look at. One thing you can look at is the coherence of the motion signals. So if you have low coherence of motion signals, like they're all kind of scattered and you're not getting a very strong motion signal in any direction. If you've got high coherence, you're getting a very strong signal. But of course, you can have a strong kind of wrong signal. So this is the bias, and you can have a bias in one direction, away from the kind of true or average trajectory, or you can have low bias. So we have measures of these kind of coherence and bias, and we reasoned that if you were going to have something that's difficult to catch, you'd want it to be kind of a strong and wrong signal. So you need a high bias, but also a strong coherence in the signal in order to make something difficult to catch. So we looked at targets that had higher than median coherence, and we looked basically at how bias then interacts with fitness in this population. And it's obviously a very messy graph. It's a very weak kind of correlation. But what is quite interesting is that where you have high bias, you do get a kind of a tendency towards slightly higher fitness, and you do get these sorts of stripy targets. So it does seem that targets that are highly coherent and also have high bias might be slightly more kind of fit in an evolutionary sense. But this obviously doesn't actually explain the results we got. So we also looked at this other measure of motion energy, which is just basically how much motion strength there is in the signal. And in this case, you get the opposite kind of response, which I think is fairly obvious, that if you have this low motion energy, you've got this sort of very grey, nondescript, kind of similar to the average background colour. And these have higher fitness than these high contrasting stripe things. So what we think is probably going on here is there's kind of two different pressures. There is this pressure of these kind of biased signals that are being caused by these, directionally misleading contours here, but actually in our particular setup, the motion energy is coming out as a stronger kind of pull on evolutionary fitness. And there are probably reasons we can think of for this to be the case. It's possible that these sorts of stripes will work better in specific situations. So you can imagine, with particular motion trajectories and particular attack trajectories, for example, maybe there is an optimal stripe orientation that will cause particular biases that will be helpful. Obviously, in our kind of fairly simple setup, they're moving in all directions and people can attack them from all directions. So maybe any effects you're getting are kind of evened out, whereas this low motion energy is a more kind of reliable. It works in a lot of different situations as a signal. So yes, this is kind of a little summary of what I've just been saying. But I think one thing that is, maybe painfully obviously missing from my discussion is, of course, this is all working on one target. So there's one little stripy zebra running about on its own, which we know is not the biological situation, right? Like this is a lot, and this is true for a lot of stripy animals. A lot do you find in herds or shoals of fish or whatever. And interestingly, this is actually relatively underexplored with motion dazzle kind of patterning and the effects of patterning on this sort of grouping. There's been a few attempts to look at it a little bit, but I think this is still not really been looked at, and it's probably because of the technological challenge of doing so in any kind of realistic way, and of analysing, like, what are you looking at here? Do you want to just see whether they can catch any target or a specific target, and all these sorts of things. But I think this would be a really interesting direction to kind of take this in a more ecologically valid way. I think it seems fairly clear to me that, at least on its own, on an individual animal, stripes are not a sort of magic bullet for making you difficult to capture, but maybe in groups they do have a stronger effect. Okay, so just to tack a little bit onto the kind of second part of my talk, which is also about kind of searching for things in a slightly different way. This is some more recent work that I've been doing with some collaborators at the University of Essex and at the University of Aberdeen. And we've been looking at kind of foraging behaviour, which I know is of a lot of interest to many people here. So what do we mean by foraging? Because it's one of these things that gets used in a ton of different ways in different literatures. So we've been looking at foraging as the decisions that animals or humans make as they are searching for multiple targets in environments. So how do they make those decisions to go from one target to the next, to the next, to the next? And the sort of classic paper that was maybe our inspiration here was this paper from the 70s by Marion Dawkins. And she was getting chickens to peck at grain. It's a lovely paper. It has some lovely pictures in it. And they basically are looking at these chickens pecking at these two types of grain, which I think are actually orange and green, but they could only do things in black and white in the 1970s, so that's how you get it reproduced. And the chicken pecks away, and what the researchers were interested in was this idea that the chickens have this template image. So you can see that the chicken kind of chooses all the black ones for a bit, and then switches a little bit to some white, and then switches back to some black. So they're not just randomly choosing between these targets, they seem to have a preference for one for a bit, and then switch to another one, and so on. And this was explained in this idea of, they have a template of what they're searching for. So this is a very classic result. And in psychology, quite a lot later, because we're a bit behind the curve, but we found, basically researchers have found very similar effects. So you can get people to forage on an iPad, and you can kind of change the stimuli and get different behaviour in participants. So if you make it quite an easy task, so for example, if you tell people just to get all the red and green targets in here, basically people don't use this sort of template. They seem to be able to switch quite easily between red and green. Whereas if you make it quite a hard task, so you say, please find the red squares and the green circles among the red circles and the green squares, then people will use the same sort of templates that the chickens use. They'll kind of go, right, I'll get all the red squares first, then all the green circles. So they favour these kind of long runs and a small number of them. And what people did was, when they were doing these papers, is they compare people's performance on this and this in terms of, the number of runs they have or something, and then they do a nice ANOVA and say, oh, there's a difference between the two conditions. Great, write the paper, let's go home. And to be honest, we thought that seemed a bit unsatisfactory. Apart from anything else, a lot of the papers use this maximum run length and number of runs, which are not even independent outcomes. If you have long runs, you have a few number of runs. And it doesn't really have any mechanistic basis. So we wanted to kind of stop throwing away quite so much information and try and understand a bit more about how the trial unfolds over time, and also that other factors could be affecting what people or other animals are doing on these sorts of experiments. So obviously one thing could be that they're following their template image, but you could also imagine other things that are affecting behaviour. So, for example, you could have one target class being more salient than the other. Maybe you have some things that are just harder to see, or maybe the chicken really prefers one type of grain over the other, and that's not incorporated into these kind of simpler statistical models. And you also have, cases where environments are patchy. So you could have, you can imagine this is one patch of food and this is another patch of food. And you could quite imagine an animal or, a forager going through here, picking up all the blue things, but this actually tells you nothing. This isn't actually a preference to stick. This is just the animal is in the bit where the blue things are and it's just going through the blue things. It's, not telling you a lot about, there are different things that could be going on here, basically. So what we have done with this is we have made a very simple kind of computational model based on sampling without replacement. So it's really just a model where all the targets could be basically balls in a bag, and you take one out and then it's gone, because that's what foraging is. So it's very simple to implement mathematically. I am not really the mathematician on the project, so I highly encourage reading the paper if you want to look at all the maths gritty details, but they are all there. But what I wanted to show you just is a kind of brief overview of kind of some of these different kind of cognitive biases that we've put into the model and how they can kind of map onto behaviour. So we have this bias for staying with the same type of target rather than switching targets, this kind of template image bias in our model. And we can actually fit that to just one trial of data. So Dawkins was working in a time when we didn't put all our data on OSF, but very nicely there's a figure of this one trial of data in the paper. So we just kind of stuck this in the model, and it can give us a reasonable fit to the data. So this is it's a Bayesian model, so it has a prior here, which just tells you that we initially had no bias. 0.5 is no preference to stick or switch, and then once you've run the model on it, the posterior comes out here. It thinks that if there's an equal number of each class of target left, the chicken will stick with the same target class with a probability of p equals 0.8 based on this data. So considering it's 100 chicken pecks or whatever, you can get a fairly tight estimate of what's going on here. And then we can add in new biases. So we've got the preference for a certain type of target A over B. In the examples we've used, we've just got 2 target types, but you could obviously extend this to 3 or 4 or whatever else you might want. And these both contribute to run behaviour. And we kind of look at cases where you can create situations where the underlying kind of summary statistics would give you completely the same answers, but our model is able to detect that the kind of what's actually going on underneath is different. So this blue example, the individual doesn't have any preference, but does slightly like to stick. In this case, this is all simulated data. They have a preference for target A, but no preference for sticking or switching. So if you just look at, the maximum run length or the number of runs, these are quite difficult to really see any differences between these conditions. And if you do an ANOVA, you don't find any evidence of a difference. But our model can quite easily distinguish these two conditions. So, you have, these are the kind of true parameter values that we know because we put them into our simulation. And so here we're recovering for the pink condition, the 0.65 and the 0.5, and the 0.5 and the 0.55. So even though they might look behaviorally similar, if you just take a very simple summary statistic, there's enough information in the data to allow you to actually see in a bit more detail why they're behaving in the way they are and what particular biases they have. And we can also, a lot of this work was going on during COVID, so we did quite a lot of looking at OSF for open science data that people have got for their papers and saying, can we see whether our model can kind of fit their data sets as well. So this is one data set we found where people were doing a foraging task with different value targets. So some of the targets had a higher value to the participants than others. And again, it's the details aren't too important, but we could see that in the case where that people had high value targets as compared to one where there was no value condition, you can quite easily pull out this preference for a particular target, which was the high value one. Another thing we added to the model was spatial layout, because this is something that's, seems intuitively very obvious and yet had been sort of ignored in all the ways of modelling this before. If you look at these runs, it is very clear that people are trying to choose targets that are quite close to the last one. They're not just going randomly across the screen. And, this is with people playing on an iPad where the energetic costs are actually quite low. In a more real world situation where somebody's actually foraging or an animal is foraging in an environment, you can imagine that spatial layout's going to be even more crucial. They're just going to go for something that's close unless otherwise, there's a really strong evidence to go to something else. So we added a proximity kind of weighting feature into the model, which is pretty much as simple as that sounds. So we include information about the distance from one target to the next. So if you imagine that you've just picked up a target here, if you had a low proximity bias, all of these other targets might still be in your kind of remit. But if you have quite a high proximity bias, you're probably only realistically going to be considering these sort of 3 or so targets around this one as your next kind of decision point. And we've also got a kind of directional bias in the model as well for essentially whether a forager continues going on in the same direction if they can, or whether they're kind of happy to backtrack on themselves. And again, this is something that we think is probably important in more real world settings, because we imagine that in a real environment, people do a lot more of this. They don't tend to, or animals, they don't tend to turn around and go back behind themselves. On a computer screen, people actually do a lot more of this. This kind of zigzagging is much more common behaviour. So kind of incorporating this into the model seemed important. And again, we simulate behaviour. So this is a simulated structured stimulus. So here, this is our kind of clumped stimuli with blue all on this side and pink all on this side. And what you can see is that the simulated observer does have this proximity bias. So the proximity weighting here basically tells you that it's got a high weighting for things that are very close by. But you can see we have no preference for one target type and no preference to stick specifically to one target type, although you might be misled if you just counted up the number of targets in a run here. So this is again helping us actually understand what's going on in a trial rather than just simply counting up these summary statistics. And again, we fit it to some real data. This is actually similar to the feature conjunction example I was showing you earlier. And you can see that, again, these conjunction trials are harder, so you have more stick bias. And you can see there's quite strong proximity, particularly actually for the feature, so the easier condition. So in the easy condition, you just pick what's closest. In the harder condition, you might start to actually look a bit further afield because you're searching particularly for a particular template item. And then this is obviously interesting, but one thing that we wanted to be able to do was look. This is obviously averaged over, multiple observers, multiple trials and things like this. You're getting this sort of posterior parameter estimates. But we really wanted to know, to what extent is the model actually any good at predicting individual target selection events? So, if you have the models giving you these weightings for the next target after you've selected this red one in the middle, and it says, right, you should pick this one because it's close and it's the same colour and all the rest of it. Is the model's prediction actually right of what people are doing here? To what extent can we predict actual trajectories done by foragers? And so we checked this, and the model does easily outperform a random chance baseline. Obviously, random chance in most of these experiments is very low because you have 20 or 40 targets that you could potentially select next. So, by the time you get to target 40, you would hope that it's getting it right. But at the beginning, it's a very low chance if you're just doing it randomly. But it is doing quite well. It's, especially on the feature tracking, it's pretty comfortably above chance and, sort of 50 to 60% of the time probably getting it getting the next target selection right. And it's quite interesting to look at individual participants. So these are just participant traces. And the top two are a couple where the model is really, really accurate. So this one on the left here, actually the model only makes one wrong guess as to where the person went next, when you kind of get it to predict where it thinks it's going to go next. So that red arrow there, the person went 12 to 13. The model thinks they should have gone to the green one labelled 35. Both of those look pretty reasonable. This is, I don't think you'd be likely to do any better than that, really. And again, this one's pretty good. There's a few cases where the model makes a different guess, but it's, fairly pretty good. These foragers are quite organised foragers. You can see they don't tend to backtrack on themselves. They, have quite a nice strategy. These people kind of look a bit more chaotic in their foraging strategy. There's a lot of sort of backtracking, and the model struggles a bit more here. So it's quite interesting to look at kind of these individual differences and how we can predict why we get these individual differences. At the moment, it seems to be, we think it's explained by differences in proximity weighting. So the model is more accurate for participants who use proximity more strongly. So people who are using other biases, it's not doing quite as well. This is probably because there's fewer of them. Most people are quite strongly oriented by proximity. So this is the reason we wanted to make this kind of model, because we think it's a good idea to try and make use of all the data. It actually needs relatively few trials. You can get quite sensible parameter estimates off, just a couple of trials, which is quite flexible for use. It's generative, so we can simulate runs of target selections. We can see how well our model can predict empirical data. And we're hoping, one of the things we're working on now is trying to build upon this model in order to actually look at kind of more complex problems. This is obviously still a very simple kind of foraging task, but we're hoping to look at kind of group foraging behaviours and how multiple animals, humans might, kind of actually forage together or compete together, possibly more accurately. And whether we can incorporate this within the same modelling framework. So that's, again, hopefully kind of an interesting kind of collective behaviour angle. So yeah, that's all I wanted to say. So just thanks to everybody who's been involved in these two projects. That's my email address if you want to contact me about anything, but I'm also obviously very happy to take questions now. Thank you.
Thanks so much, Anna. That was wonderful. And we're taking questions from the room.
Thank you so much. I was just wondering, can that model have more than 2 types of options, like 3 or 4 different types of patch?
Yes, yeah. In principle, absolutely. We haven't done it because we started to get really confused with the indexing, but it's not, that's just, the maths is very straightforward. Yeah, we've done it with 2 because that's, most people so far have used 2 types of targets, but for sure it can be extended.
Thank you.
Thank you. I was wondering whether response time matters in determining sticking and switching, and whether this could potentially explain individual differences.
Yes, I think this has been looked at. I cannot remember the details of it off the top of my head. We haven't included response time in our model. We do have that data. We've just included the order of target collection. And we've kind of assumed that distance is almost a proxy for response time. Not so much in these sorts of on target, selection games on an iPad, but I think certainly in more real foraging tasks, it would be a decent proxy. It could. I think people do forage slower in those sorts of conjunction, more difficult conditions, because they've got this template that they're searching for. And therefore so it certainly could interact, I think. I think it depends, what you think is the kind of base process going on, in a sense, whether you think that the response time is underlying some other kind of cognitive processing that's having to go on. And therefore you can see it as like a downstream measure almost. Yeah. But yeah.
Thanks.
Yeah, thanks. Was super interesting. What happens if you like in like modeling foraging?
Yes.
One parameter that you didn't talk much about, but other people have used is the like the amount of reward per item. So like some foraging models think in terms of like reward landscapes, and you like you invest energy, or you invest, or you there are risks along the way, because you might be running in targets which come with a penalty or something. And so like I remember these experiments by, I think, Larry Maloney at NYU. We did lots of that kind of stuff. How would that feed into your model? So if you say you've got two different classes of targets, and they come with, or you've got targets which are positively rewarded, and other targets which come with a penalty or something, how would that?
So at the moment we don't. So at the moment we have this kind of class bias where you could have, if you have a static, if you know in advance that this target is worth more than this one, that the model can kind of capture that. And perhaps you might have a preferred type of food or whatever. If an animal or a forager does, the model will pick that up. So it can kind of deal with that sort of intrinsic or extrinsic, but stable preference. What it doesn't do at the moment is learning. So you can imagine a situation where, we were talking about this earlier, but you don't know yet whether the red or green ones are good. And then you find out as you go through your foraging task that actually the red ones are great and really tasty, and the green ones actually make you feel a bit ill. So you kind of update, as you're going along, and you might turn away from the red ones, right? Which is this kind of. So I think that is perfect. We could definitely put in a time-dependent parameter for this kind of bias, this preference. At the moment it's just static, and that's again partly because of the sorts of experiments we've used where that's just been known up front. The participant knows whether there's a value or not associated. But I agree, for a natural situation that would be a really interesting kind of extension to the model, and make it kind of more usable in a wider variety of domains.
Yeah, thank you very much, Anna, for this fascinating talk. I would like to go back to the motion dazzle.
Okay, yes.
And would like to ask the question, so you as an ecological limitation, you pointed out that it may be that there's other individuals around and not only a single one. But another limitation that I saw is, of course, the two-dimensionality that you have.
Yes, of course.
So do the stripes help you maybe in a three-dimensional environment, so that it's something about the depth perception or something like this? Has anyone been doing these kinds of experiments? I know two former collaborators of mine, Bettina von Helversen and Lael Schooler, have also done this in a two-dimensional setting, I think with touch screens, catching snakes or something of the like, but I think it was also two-dimensional.
Yeah.
So should we do this in VR, in 3D? Would it be worthwhile?
I think for sure, I think genuinely yes. There's been very little on this. I've seen a couple, there is a paper out there with somebody doing locusts and kind of looming stimuli and this sort of thing with different stripiness and all this sort of stuff. And I've seen occasional, sort of everybody always agrees, we should do a VR experiment. I think what gets challenging is just doing it in a way that's controlled, but also, sort of valid in some way that's interesting and ecological. I think everybody kind of stumbles possibly at the publication process, because it's very easy for any reviewer to say, oh, well, the motion's not like you might expect it to be. And there's, I think there is stuff out there, but it's, maybe we should just all be pre-registering it and putting it on OSF anyway, but just to show we tried this and it didn't work. But yeah, I think that's the obviously 3D, you're absolutely right. There's some really interesting, when you think about, especially with like zebras, the way that the zebras will interact with there's a very stereotyped attack motion, right? So you can imagine that there might be ways that zebras could have evolved to exploit that, but it's a very specific 3D arena. And I think that would be very cool to look at, actually. You know, I'm not sure that my participants would love me doing, you know, now you must run towards this herd of moving zebra. We're going to get some very tired participants. But, you know, that's, I think, you know, maybe a good way to go with this sort of thing. Yeah, for sure.
Could you apply the models that you have used in a first step before doing human experiments on this?
Yes, I think basically the answer is yes. I think people have done these kinds of models in 3D environments. It all gets even more complicated because it essentially becomes 4D. But it's, I think it would be for sure possible to do that. And people have looked at certainly the motion component of it. And then really all we're adding is whether the patterning condition that you've been allocated is varying anything. Yeah. Thank you.
Thank you.
Any more questions? Now's the time. Yep.
Thanks. Maybe you already said it, but I may have missed it. Are there some hypotheses for the differences between individuals in their foraging?
These kinds of thoughts. Yeah. So I think in this case, what you're looking at is the model is very good if the people are mostly focused on proximity, and is not yet as good at these people. If you actually look at their patterns, they're people who are really strongly foraging in runs. So they're really selecting all of one target type and then all of another. And what the model is less able to do in those cases at the moment is figure out why they've gone to a particular. It doesn't seem to be quite as strongly proximity based in these cases. It probably still is to some extent, and I think these people are possibly a little bit more chaotic even than that. One other thing that we're interested in is a kind of planning component, because everything at the moment in the model just looks one target ahead. But what you might imagine people do is they start off up here and then they go, oh, there's actually a good little cluster over here. I'll just make a big jump and go over here and kind of sort that bit out, which the model wouldn't necessarily anticipate. It might say, no, you should stay closer because, you know, there are still some targets here. But it's almost like a patch leaving thing, right? Where you're like, oh, there's maybe 1 or 2 still here, but actually there's 10 over there. So I would rather go over there and start exploiting that bit. So that may also be something that's different.
So there were human participants?
In this, yes.
Okay.
Yeah.
And did you ask them to describe their...
No, so this is all secondary data analysis. So this is data that was collected in, well, a long time before that, actually.
Oh, yeah.
So unfortunately, we couldn't ask, but it would be interesting to ask people more about what they think their strategy is and whether they just go, I was bored and wanted the course credit, you know.
Thanks.
Thank you.
Yep, sure.
Thanks for your talk. I was wondering if your foraging model could also be applied to study foraging strategies for like animals that use other senses to forage.
Oh, a very good question. Yeah, this is obviously very visually based. I think in principle, yes, absolutely. I think to work out the details of how to do it. But yeah, you know, certainly the biases you could, you know, do by could be 2 different target classes of different odors or whatever. Proximity is obviously also going to be very important. Yeah, I'd be interested to know whether something like the stick or switch kind of template is also important. I don't know if we know about that, but it would be really interesting to find out if that is also something you see, that, you know, they tend to forage in runs. So yeah, I think the model is obviously not actually seeing anything. So I think any type of target sensory modality could go in there.
So thank you very much, Anna. And unfortunately, Anna is leaving us today already. She's been around for almost a week, and we hope she'll be back sometime. So
if you think of any burning questions, please feel free to contact me. As I say, we're kind of interested in, if you've got any aspects that you think might be interesting collaborations or anything, just drop us a line. And very happy. We're very happy to take random data sets you have in a file drawer somewhere that you think might go into a foraging model. We're very keen for that.